Khan, a neuroscientist at the University of Tennessee. He's digging deep into the gut-brain connection and the neuroimmune pathways. His current research is funded by the DoD, and he's helping to understand how CTNNB1 may impact the body far beyond the brain. Please welcome Dr. Khan.
Okay. So good afternoon to everyone. I'm a basic neuroscientist at the University of Tennessee Health Science Center. And I'd like to thank Effie and Emily for giving me opportunity to speak today and the kind invitation over here. So today I'm going to talk about the gut microbiome in the CTNNB syndrome. So why we focus on the gut microbiome in the brain disease, which is the peripheral. All the neurological disease, we focus on the brain-centric mechanism. Why we focus on the peripheral mechanism? That's what I'm going to discuss in the next few slides, and why we choose to study on the gut microbiome in CTNNB. I'm going to provide the rationale and logic and the data we generated with the few samples I'm going to present today. So the gut microbiome is, the CTNNB is a monogenic neurodevelopmental disorder. And like many other brain diseases or genetic diseases, environmental factors play a key role in the disease onset and the progressions. Gut microbiome is one of the environmental factors and has been shown to play a key role in the in maintenance of the human health and the disease pathogenesis. Unlike the genetic factor, the gut microbiome can be modified by the diet and the other interventions. This gut microbiome actually harbor in the gastrointestinal tract, and it can modify and educate immune response, host behavior function, and the brain function through the gut-brain axis. So I'll begin the slide with the. Sorry,
it's not showing. Are you changing?
No, I'm just going to basic. Sorry, I just wanted to give about the brief introduction about the gut microbiome. Okay. So we all know the CTNNB syndrome is a rare neurodevelopmental disorder which are affecting children worldwide. The disease is caused by the de novo heterozygous mutation in the CTNNB gene, which code a beta-catenin, which is very important protein playing a role at the gut epithelium integrity. The prevalence of disease is 2 to 3 per 100,000 births. The disease is characterized by the developmental delay, intellectual disability, and speech impairment, and the other impairment, like just previous speaker talked about the vision impairment. Currently, no disease modifying therapy exists. One of the major barriers for the development of therapy is to understand the mechanism underlying the disease. And here we bring that the gut microbiome is one of the mechanisms. The potential mechanism has been discussed and published that show that beta-catenin and the Wnt signaling is playing a role in the CTNNB syndrome. I'm not going to talk about this mechanism because the previous speaker and the following speaker, they are going to talk more detail about this signaling. But the idea is this signaling is responsible for the impairment of the transcriptions, which make a protein, so RNA level dysregulation and then make a protein is affected, and this all is responsible for this intellectual disability and neurodevelopmental deficit underlying the CTNNB syndrome. So when I started working on the gut microbiome in other genetic neurological diseases, which started during the childhood, so one of the things come up, is there any other mechanism other than the brain that could play a role? And then we come up the gut microbiome. Dysbiosis could be a hidden driver for the neurodevelopmental deficit. The reason is the gut microbiome is actually shaped the early brain development. During that, the window is very important where the gut microbiome shaping the early development. Is there any therapy that could target the gut microbiome? Could be a promising new therapy for the patients which are affected with the genetic disease that could be a safe, effective, no side effect. So then I always say there is a discussion between the organs of the gut, liver, brain, and the heart, who's the most important organ in our body. And then I'm a neuroscientist, I'll say, oh, of course, brain is I'm the boss, I control everything. Then heart say, okay, without my beats, you are all done. So every organ has a rationale, like liver, I clean up your messes, keep the system running, and I deserve a raise. And the gut was listening quietly, okay, you guys are talking a lot, ignoring me, okay, I'm shutting down. And what happened is a few miserable, bloated, and cranky days later, total system crash. So from the diet, everybody say, okay, gut is important. And you say, you might have observed every speaker at the end of the slide, they say gut instinct. My gut instinct says that one. So from the day, everybody agrees the gut is playing a role. So, and the gut concept, the gut microbiome concept actually is coming from a long time before, when it says that all disease begin in the gut. And gut is also considered as a mini brain or second brain. These are the few examples that could let you think about that human body hosts 40 trillion bacterial cells, which is outnumbering the approximately 30 trillion human cells. There are 100 times more microbial genes than our own human genes. Our genes turn and off in response to what the microbes do, and vice versa. So gut is a multitasking. It's not only playing a role in only the gut gastric problem, but also playing a role in the hormone productions, immune response, as well as neurotransmitter productions. So why we focus on the gut microbiome aspect in the CTNNB syndrome? CTNNB, which code the beta-catenin, is one of the important protein in the gut epithelials. It's part of the adherence and the tight junction protein which maintain the gut barrier integrity. So one can assume if there is any dysfunction in the CTNNB or beta-catenin could impact the gut barrier integrity. And there is a connection between the gut and brain through the circulation, through the vagus nerve. So any dysfunction in the gut could have an impact on the brain. Gut dysbiosis is a common feature across the spectrum of rare neurological disorders. So I'm going to give a few examples. We are working in our lab is a neurological genetic disorder where we identified that the gut microbiome actually is playing a role are contributing the disease onset. One of the disease is called the DYT1 dystonia, is a primary childhood onset disorder caused by mutation in the TOR1 genes. And the mechanism is the proposed mechanism is the brain dysfunction is causing, the mutation in the DYT1 is causing the brain function abnormality, which leads to the abnormal behavior. We identified that the gut microbiome at certain point during the early stage of disease is causing a problem. And then we assess the gut microbial richness through the 16S ribosomal sequencing. If you see the graph, there is a clear separation of the microbial diversity between the wild-type mice, that's a control mice, healthy mice, and the other one is the TOR1 where there is a mutations. And these gut microbiome composition or diversity is correlated. If you look up the bottom panel, that's the behavior function we quantify into the mouse model. And then what we do it, okay, there is a microbiome diversity among them. Is there anything in the gut barrier integrity? So we took out the gut from the mice, from the healthy mice and the dystonia mice. And we see, you see the circle, the yellow circle, the beta-catenin is downregulated in the gut. That shows there is a beta-catenin dysfunction, disrupt the gut barrier integrity and release the gut microbiome metabolites into the circulation that could impact the brain function. So other disease is a PNKD, is kind of dystonia where you can see the difference between the wild-type mice. This is the normal mice that walk around in the cage without any obvious symptom, just enjoying the life in the cage. Okay, due to the time scarcity, I'm going to cut down a little bit video, and then I'm going to show you the other mouse which have a dystonia symptom. They have a mutation in the PNKT. If you see the mouse, it shows the dystonia dyskinesia symptom. Hind limb and forelimb is clearly showing the disability in their mice. Okay, I'm sorry about the timing issue, so I'm going to cut down the video. Okay, so the mechanism, recently we identified a mechanism in the gut microbiome dysfunction in this mice. It has never been discovered that the gut microbiome has been playing a role in the PNKT mice. And we see there is a microbial diversity between the healthy mice and the mice which have a dystonia and dyskinesia symptom. And here in this mouse model, again, you see the red circle is the CTNNB genes that encode the beta-catenin is downregulated or disrupted. So is there any correlation between the CTNNB and the gut microbiome? So we started working on the three major goals. One is to identify the association or cause and effect relation between the CTNNB and the gut microbial signature. Discover the microbiome-based molecular pathway that we could target and identify and test the novel therapeutic strategy, including the prebiotic, probiotics, and the youthful and healthy fecal microbiota transplantations. So starting with the first goal, we got the fecal sample from the CTNNB Foundation. We isolated the DNA and then subjected to 16S ribosomal RNA sequencing. This is the way we do it when we get the fecal sample, we isolate DNA and we do the sequencing. The data is very surprising. So far, I've never seen like n is equal to 5. We do see a significant changes in the microbial diversity. We had only n is control, n is equal to 5 for the controls, and the one is the CTNNB. You see these are the alpha diversity index that indicate the microbial richness and diversity. The CHA1 and the Shannon show there is a trends of microbial diversity. Although it's not significant, I assume the sample size is very low, but the other index is called Shannon that show a significant even with the low sample size. So that data clearly show there is a microbial diversity between the CTNNB patient and the control. And these control are from the isogenic controls, they are from the siblings. Similar to the alpha diversity, we do the beta diversity, and you see the principal component of analysis reveal a 42.54 clear separation between the genotype, between the control and the CTNNB patient. And then we identify the bacterial profile from the phylum, from the family, from the genus, and from the species. If like you are going to United States, then you go to the state, then you go to the city, then you go to the town. The same thing, the gut microprofile, we do the family, phylum, family, genus, species. So these are the bacterial we identified. Some bacteria are very good bacteria which are depleted into the CTNNB stool samples. And these bacteria actually produce the butyrate, which is responsible for the gut and brain functions. And these are significantly downregulated into the fecal. But again, this is very, sample size is very less, so it's very premature conclusion. So these are all the gut microbiome profile which are significantly altered between the control and the CTNNB stools. We identified it. And based on this one, we did the functional prediction analysis, and there is a few pathways that are altered. Some of them are related to the energy metabolism and oxidative phosphorylations. Some are vitamins related, some are lipid and sulfur metabolism, some are related to that give a hint that is related with the neurological diseases like Parkinson disease. So with all the three goals, we achieve half of the first goal, like there is association between the CTNNB and the microbial signature. But we don't know whether it's a causative or just is just association between. The next goal we are working on to identify the microbiome-based molecular pathway. And the third, once we identify it, then we test the probiotics or identify good bacteria, transplant into the mice, CTNNB mice, and see if there is any effect on that behavior functions or the brain function. So the ongoing study we are doing in our lab is what we do it to establish the. We have 1 minute. That's the last slide. Okay. So what we are doing it, we already showed the association between the microbial signature and the CTNNB. So this one data we are generating it whether there is a causative effect of the CTNNB on the behavior function or the brain function done. So what we are doing it, we took the stool from the control healthy subject and from the CTNNB patient kids. And we inject the CTNNB stool, human stool, into the mice which doesn't have a microbiome. So we deplete the gut microbiome with the antibiotic cocktail, so there is no gut microbiome in their guts. So if the CTNNB stool is a pathogenic, if it goes into the mice which are germ-free, it should, if it's pathogenic, then it's called the behavior functions or something brain function. That is the study we are going on it. So far we have generated the data on the microbial signature and the CTNNB mutation. And that's all. And then I'd like to thanks to the funding agency and my lab member who generated this data, and of course the CTNNB Foundation. They provide the fecal samples to study on this new very innovative direction on the CTNNB syndrome. Thank you, and thank you so much.
Thank you. Can you hear me? Okay, thanks, Dr. Khan. I might just rehire GI. I gave up on him, but with you, I might rehire him. Okay, we have a few minutes for questions. I'm going to push it just a little into our break, if that's okay. So if you really need to go ahead and feel free. So if I can get Drew and Ramin up here, you guys can cozy up and we'll have a couple questions. I'm going to ask one real quick for someone whose child is in evaluations. Ramin, her son is 9 and was diagnosed with tethered cord syndrome, and her doctor is recommending that she doesn't get a release surgery until he hits another growth spurt, and she's wondering if that's where she should push back or if that's fine. Oh, yeah. Right there.
9 years old, you said?
9.
So I would say that the majority of kids with tethered cord are going to present with signs and symptoms well before 9. However, we do counsel patients and families that if they truly have tethered cord, doing it before the big growth spurts is actually recommended. And we have seen some reversal of urological function or dysfunction even at age 10 or 11, but it becomes really hard to get some of those things back once you've had nerve injury that long.
My question is for Dr. Scholes. So my son did have the fever screening and was found to have stage 1. So my question is there any idea of timeline through the progression of stages? If he has stage 1, I know you said he can stay through it, but if he does go through the stages, is there any timeline? And also, is it possible that they can have different stages in each eye?
So thank you for the question. The second question is much easier. So fever is classically asymmetric. So there can be children with stage 5 in one eye and stage 1 or nothing in the other eye. So that definitely can happen, including in CTNNB1. For kids with CTNNB1 in early stage disease, so like very mild stage 1, I personally recommend repeating the screening probably annually or biannually for many years to see where it's headed, because I don't know. There are many kids with fever at large who have stage 1, like I mentioned, and it just stays stable forever, but we don't know if CTNNB1 will always follow the same pattern or it'll do something different. So you just have to keep watching.
Hi, my name is Charlotte. I am a parent to Grayson. We are new to the community. He is 13. We've been seeing an eye doctor for a long time. My brain is on overload, like a Dad said earlier today, and I'm curious if once you have the CTNNB1 diagnosis, we should be seeing a specialist to have the fluorescein done, because that has never been part of our conversation with our eye doctor.
I think yes is the answer, because from my work and from the group in Utah, there are many kids who are seen for years and in clinic, and sometimes the participation is good, sometimes it's not, but you will miss subtle eye disease unless you get a fluorescein. Usually that requires sedation to get the best data.
My question is in regards to retethering. Do we know if there is any incidence of that, or what's recommended as far as after tethered cord release surgery, what the next steps should be?
That's a great question. So it sort of depends on the reason for the tethering. So if it was a very straightforward detethering operation, which sort of that phylum cutting, which is a very common surgery, the incidence of retethering is really low. I would say less than 5%, maybe even less than 1%. I think that the cases of retethering usually, again, present during growth spurt. And so if you do notice that there are signs, especially if there were signs before, if those same signs are coming back, I would definitely make sure to get imaging and also follow up with urology. And if they're having physical therapy, get an objective physical therapy evaluation. Sometimes we are not the most objective, especially if we did the surgery, we don't want it to be retethered. So getting objective evidence is really important.
This question's about fever. My daughter had the exam under anesthesia before you came into our world with all your amazing documentation, and it was very difficult to get the doctor to even do the exam under anesthesia, so thank you. But after that, it was found there was no signs of it, and I was told we would never need to test her again, and he's been adamant about not retesting her again. She's now 6. This exam happened around age 3. What would be your suggestion, and is that something I should push for, or do you, is there a time when you're in the clear? I know that's a black and white question. That's probably not possible, but.
Yeah, it's hard to be 100% sure in every case, like you alluded to, but if there's really nothing, then it's hard to know if you have to repeat it or not. A parent asked me the same question at lunch today, and I think that if it's totally normal, repeating it at one point again in the future, non-urgently, is probably enough, like 2, 3, 4 years later when there's some other anesthesia for something else. But I don't know if that's actually necessary. I'm just not sure, and I would hate to miss something. That's a reasonable time to look, yeah. Mm-hmm.
Okay, let's have one more question. Who has it?
Hi, I'm Anita. I asked a question earlier, sorry. But I have an 18-year-old, just turned 18, and really did the fight for trying to get them to understand the fever. And so now I have more information to give to them. They did all of their eye checks. You know, she's been seen by a ophthalmologist her whole life. So I guess 2 questions. One is everything that you had said about retinal specialists, everything's pediatric. Just move it to an adult. Is it relevant still at the adult stage, because she's now 18 and she's never had the fluorescein?
I see. You might be better off, since you're just at the cusp, seeing somebody who's willing to see kids. Not to say anything different about adult specialists, but I think Ramin could agree with me that even though a patient's 18, if they have neurodevelopmental disorder, sometimes it's a little bit better suited to a pediatric specialist. But everybody's different. Yeah.
Okay, one more question actually for Dr. Khan.
Hey, Dr. Khan, thank you for being here. Just a question about gut health in our disease population. Do you recommend probiotics? Do you recommend, sorry, getting feedback. Do you recommend seeing a nutrition, like what would, I know you're not a physician, but what would you say for kind of monitoring gut symptoms in our families?
No, that's a good question about the probiotics or prebiotics. I recently met one patient with the disease. We are working on it, and the kids are diagnosed with a childhood onset neurodegenerative disease. And then she was telling me, And unfortunately, there is no cure for that disease as well. It's like the CTNNB, many symptoms overlap with that disease. And the mother was telling me that they are giving the yogurt, to the kids like almost every day. And then the kids are feeling very well, is the symptom. So at this stage with the CTNNB, yeah, definitely the probiotic is going to be helpful. It's not going to be a therapy, but it's going to be helpful with the symptom. But once we establish there is a causative effect of the CTNNB in the behavior functions or the brain function, then we could come up with idea that maybe probiotic or prebiotic or anything with the youthful bacteria that we could identify can be given as a dietary supplement. If you go to Costco, you see the probiotics, many bacterial in the probiotic, lactobacillus, pneumococcus, these are all inside the ingredients of the probiotics. And these are all altered into the CTNNB stool fecal sample. So definitely is a good starting point on that.
Thank you.